have set benchmarks in clinical excellence. Shri Ram Cancer and Super Speciality Center, affiliated with the Mahatma Gandhi University, stands as the state's largest multidisciplinary oncology facility equipped with cutting-edge technology like robotic surgery, cyber-knife radio-stereotactic surgery, renal accelerators, 3 Tesla MRI scan, PET scans, etc. to name a few, and is managed by eminent faculty like Dr. Hemant Malhotra, the director of the Shri Ram Cancer Center, and his very capable team of colleagues and doctors and scientists. The center's comprehensive services include advanced radiation oncology, bone marrow transplantation for benign, malignant, and thalassaemic conditions, and a state-of-the-art blood bank that is accredited by American Association of Blood Banking and NABH. Similarly, the Center for Neurosciences and Cardiac Sciences, led by a team of very competent doctors, performs complex procedures using the latest equipment, further enhancing the university's reputation as a hub for advanced medical care. With recognition from National Medical Commission and the Government of India, the university offers a diverse range of undergraduate, postgraduate and super specialty programs, attracting students seeking quality education and healthcare professionals dedicated to excellence. Through partnerships with the state healthcare schemes, the university ensures universal access to high-quality healthcare, emphasizing a commitment to service, quality and patient-centered care. I most humbly invite all our esteemed guests to take a tour of the hospital and witness its services to the society firsthand. The visiting foreign dignitaries this morning today already had a brief tour of the Mahatma Gandhi Hospital and the Shri Ram Cancer Center and the Center for Cancer Immunotherapy. The theme for this year's conference emphasizes the synergy between translational science and immunotherapy, highlighting novel strategies that bridge fundamental research with real-world clinical applications. Over the next couple of days, we look forward to engaging discussions, insightful deliberations, and pioneering research presentations that will reshape the future of cancer treatment. We at Mahatma Gandhi University are committed to fostering an ecosystem of scientific inquiry and collaboration. As the host institute, we take immense pride in facilitating this academic congregation and hope that our time here will be both intellectually stimulating and enriching. I extend my heartfelt gratitude to all our distinguished speakers, researchers and participants who have traveled from different corners of the world and this great country to be here today. May this Congress be a symbol of sparking innovations, igniting new ideas and collaborations that I am sure will drive forward our collective mission of eradicating cancer. With this formal inauguration of the 4th World Congress on Translational Cancer Research and Immunotherapy, I wish you all a highly productive and successful event. May God bless us all. Thank you.
Thank you, sir. We are honored to have with us the founder and emeritus chairperson of Mahatma Gandhi University of Medical Sciences and Technology, Professor of Obstetrics and Gynecology, and renowned specialist in reproductive medicine and IVF technology, founder of the first IVF Center of North India, Dr. M.L. Swankar. A visionary leader, Dr. M.L. Swankar aspires to make this institution a pioneer in the field of medicine and research. He is constantly promoting and encouraging an environment supportive to medical advancements and translational research. The establishment of the Center for Cancer Immunotherapy at Mahatma Gandhi Medical College is an evidence of his commitment towards the need of translation of scientific research from bench to bedside. I humbly request Dr. M.L. Swankar to address us all on this occasion.
Thank you, Dr. Mishra. सभी को मेरा नमस्कार. Good morning. First of all, I express my gratitude to Nobel laureate Professor James Allison, chief guest of this function, father of immunotherapy. We are very lucky. It is the blessings of Almighty that he is visiting our university. So we are grateful to you. Your presence here will give inspiration to our faculty, to our students, and those who are taking training in different.
Work towards the end, provided by Naveen Sharma and Stephen Mott. And all of this, of course, has been done by my colleague and partner in science and in life, Dr. Patrick Sharma, who really has helped me learn the clinical ins and outs of what we need to do, a process that I'm still learning about. So just to review some basic immunology, immunotherapy, T cell is all based on T cells. T cells were discovered actually when I was in graduate school, a little bit before I was in graduate school, which dates me, I'm sure. But when I heard about them in a course, all that they really knew was that there were these cells that could kill virus-infected cells. They go through your body, percolate through your tissues, through your blood, through the lymph, and basically survey for infections. At the time, I asked the professor, how does it work? How do they recognize things? And he said, we don't know at all. He was a skeptic. He didn't think there was such a thing as T cells. He thought they were just macrophages that had an antibody stuck on their surface, and that was it. But anyway, so we began to work on it. The T cell antigen receptor we identified in the early 1980s. It's a dimeric molecule, but we thought, and everybody thought at the time, that it was a binary thing. You just flip that switch and it turned the T cells on. It turned out that was not the case. Very quickly it became apparent that it was more complicated. There was a second signal called a co-stimulatory signal that we showed in 1988 was mediated by a molecule called CD28. And until the T cell got both of those signals together, and this is a critical important thing that I'll come back to in a moment, the T cell must get simultaneous delivery of both those signals to get activated and start all that stuff in green there. I'm not going to go through it. But to amplify and expand an individual T cell, you've probably got about 10 to the 10th power different T cells with different receptors in your body, which means a very small number of them can do anything about any given antigen because they're all specific of one thing. So you need to get an army, and that's what this first step is to tell that cell, don't just sit there and cruise around, but divide as fast as bacteria for a week or so, generate an army that can take care of whatever the problem is, and then it's got to stop. And so that's a complex process. But the first step, as I said, is contemporaneous engagement of these two receptors, the antigen receptor CD28. But we found in the early 90s that there was another molecule involved, CTLA4, a very mysterious molecule at the time, which was very homologous to CD28. It's not expressed in resting T cells. It's only induced after activation. And its function, as we found showed, is to come up, accumulate, and actually inhibit, we think, by outcompeting CD28 for its ligands on dendritic cells. The ligands for CD28, I should point out, are found exclusively almost on very important cells called dendritic cells that Dr. Bardwaj and Dr. Murad are experts in studying to get the whole thing started. Other kinds of cells can't provide that signal. But anyway, the T cell gets going, expands, and then CTLA4 stops the proliferation because otherwise your T cells will kill you. When Tac Mac and Arlene Sharp and Cynthia Chambers in my lab knocked out CTLA4, we all found that the mice die when they're about 3 weeks old because their immune systems are out of control and they just kill them. So this is a hardwired molecule that is required to actually protect you from destruction by your own immune cell. Just to show you how these things work, this is CD28. These are fusions that we made of CD28 and GFP. That's a T cell coming in contact with an antigen-presenting cell. And you can see the false color image. The red is just the CD28 coming together in the synapse where the T cell receptor got a signal. That tells the CD28 in the cell to come in and help provide and amplify the T cell receptor signal so the T cell gets activated. CTLA4 is very different. It's made in these little vesicles. It's made in the back end of the T cell. It's there. The T cell is cruising around. As you can see, let me start this again. You can see the back of the cell. And the CTLA4 is drugged. It's a little vesicles tied by microtubule filaments. to the microtubule organizing center, and as the cell polarizes, it drags CTLA-4, sprays it exactly into the synapse, and we think it pushes CD28 out of the way and just stops the T cell signaling and turns off the expansion. In any event, we did a lot of work in mice, as the panel showed on the right here. This just shows what happens after it's in the synapse. We did many experiments of the sort shown on the right. I'm not going to go through it because they essentially all look like this. You inject a transplantable tumor on mice, syngeneic tumor, and the tumors grow. You have to euthanize the mice. If you block CD28 with an antibody, the tumor actually grows faster, showing there is a nascent immune response, but it just can't get the job done. But when we injected anti-CTLA-4, you can see the tumors grew for a while and then went away. And the tumors then, the mice were immune to re-challenge for the rest of their lives. And this happens because there is no there is no B7 on tumor cells, and so the tumor cells have a head start until they get to a point where they start dying and they activate the innate immune response and get the dendritic cells in there. The T cells are of no use until they get triggered by the dendritic cells. Then they can come back and kill the tumor cells. So that's what that was all about. And as soon as we saw this result, it became apparent that this could be a really interesting thing to do. First of all, because you're not treating the cancer, you're treating the immune system. The target is on the immune system. So this could potentially work against any kind of cancer. All cancers, they're caused by mutations. They are going to have antigens generated by mutations that are not there in normal cells. And if they generate peptides, as the MHC molecules can present, they become targets. And so it doesn't matter the kind of cancer. The T cells really don't know in the first approximation what kind of cancer it is, where it came from. They just know there are things on the surface that are different. So it has the possibility to be a universal cancer therapy. The second thing is, since this whole process starts with tumor cell death, priming it, you could use radiation in combination. You can use chemotherapy in combination. You can use surgery in combination. Anything which kills tumor cells would start this process. It would normally be stopped by CTLA-4 and not allowed to go, perhaps, to the whole point of eliminating tumors. But we could add CTLA-4 to radiation, to chemo, to surgery. It is being done now to amplify those responses, give those things the properties, the universality of the immunotherapy. Plus, the most important thing is memory. I'm going to start on this, I'm going to finish on this. The memory is the important thing because the antibody is just a prodrug. It's just something to get the T cells going. The real drug is the T cell, and that is a living drug that's with you potentially for the rest of your life. So if the tumor comes back, the immune system's there to take it out and protect you. And you can, if the process happens, you can start it over again and get new cells created. So does it work? Well, I show this picture because this is the first patient I met that had been received this drug early on in 2004. In 2005, she had failed already all the therapy she got. She was 22 years old, just finished college, was engaged to be married, about to start her career. She was an athlete, couldn't breathe, went to her doctor. He determined that she had tumors in her lungs. She came to slow cataract. She had 31 metastases in her lung. Her liver was totally riddled, too many to count, metastases in her liver. She had a 1.5 centimeter brain tumor and failed every treatment, including radiotherapy, to the focused radiotherapy to the brain tumor. Failed all that and was hospice bound. Dr. Jed Walchuk, her oncologist, said, "You'll try this new thing, ipilimumab. It's the last thing I've got. It's all I've got." And he gave her an injection. A few months later, she came back, was scanned. The radiologist reported no cancer anywhere, not in her brain, not in her liver, not in her lungs. She was a complete responder. This CAT scan on the right shows, I can get the mouse. This shows some of the mets in her lungs that she was completely tumor-free. At that time, I met her. They actually were it was so unexpected that they first thought that they'd mixed up the results from the scan and gotten another patient or something because there was no cancer, but it turned out it was. Anyway, I met her then. That was in 2006 when she completed the story. This was 10 years later with her 2 children. And I can tell you she's alive and well today. Her children are about to start college. We became good friends, and she went with Dr. Sharma and I to Stockholm for the Nobel Prize ceremony. There's Sharon and I, basically 12 years after she had the therapy. And as I said, she's still alive today, living her life. So, and I hope for more like that. Anyway, so this was the results of a phase 3 trial of about 800 patients, randomized trial. At the time, there was no standard of care for metastatic melanoma. As you can see here, the median survival was about 7 months after treatment, and virtually no one was alive 5 years after therapy. And so that's shown by this control curve here at the bottom. This curve, the dashed one here, that's the IPI-only trial patients. You can see it moved the median survival over about 4 months. So that would have been sufficient for approval at that time, because there was nothing else that had ever done that. No drug of any sort had ever prolonged survival in melanoma. But the cool thing is that you can see the tail of the curve flattens in about 2 years and stayed there for the 4 years that this trial ran. The endpoint of this trial was overall survival. So, but you can see that about 20% of the patients were alive in 4 years. Shortly after that, BMS and Medarex had enough data on almost 5,000 patients that had 10-year survival. It showed that 10 years, the survival was 22%. Again, it stayed. So after about 2 years, no one died. Virtually no one died from melanoma after the therapy. And that's a single round of treatment. There was no recurrence, and they were not retreated. So that was quite a success at the time, but it's 20%. Why isn't it 100%? Well, I could give you a lot of mechanistic reasons. Maybe that priming event is not going on at the time the drug's there, or something else. But of course, the more interesting thing is maybe there are other checkpoints, because this is the first cell-intrinsic T-cell checkpoint that was identified with CTLA-4. But as our first phase 1 data were coming out, by the way, 3 patients in the phase 1 trial, 3 of them had complete remissions, even in the phase 1. But anyway, as those data came out, Tosco Hanjo and his collaborators, Arlene Sharp and Gordon Freeman at the Data Pharma, showed that there was a molecule that Tosco had been working on for years called PD-1, was actually another checkpoint. It has a couple of ligands. One of them is expressed on tumors in response to T-cell attacks. The T-cell comes in, it sees an angiola tumor, makes gamma interferon. The tumor responds by upregulating the ligand for PD-1, which then turns off that T-cell, another way of inactivating T-cells distinct from that of CTLA-4. This is something that is a protective mechanism that's been shown to be involved in protecting the developing embryo from destruction by the maternal T-cells going after paternally encoded antigens on the fetus. So this is a normal molecule to protect cells that tumors have co-opted apparently to protect themselves. In any event, antibodies were prepared against that. It also, as CTLA-4 did, worked in many different kinds of cancer. But again, concentrating on melanoma, these were put together, and Dr. Walchuk and others had a combination trial, and this shows the 10-year follow-up right here. As you can see, this is the CTLA-4 only, PD-1 only. The combination, 52% of patients that are alive 10 years, again, after a single round of treatment. And so they're cured. We can use the word cure, I think, based on these sort of data in many patients, with the word cancer in the same sentence. And so, luckily, it wasn't just melanoma. This is a very busy slide, and I apologize for that, but the main point I want to make here is there are a lot of approvals of CTLA-4, PD-1, mostly PD-1, or combination antibodies now in a lot of different indications of cancer. Those melanoma, lung cancer, bladder cancer, head and neck cancer, these cancers that are caused by obvious carcinogens, in the case of melanoma, UV radiation, in the case of the other, smoking and things, that will respond very well. Other cancers don't, and that's related to the mutational burden. This shows that kinds of cancer differ in their mutational burdens. And those that have the highest mutation loads respond best, shown here in the curve. When you calculate, according to the frequency of cancers, different cancers in the U.S., we're reaching, actually this is a little bit out of date now, we're reaching about 25% of all cancer patients with objective responses due to checkpoint immunotherapy. And that's improving with time. It's moving earlier and earlier and being used in neoadjuvant studies. And so, and we're continuing to develop it for more patients. But it's important, I think, as we move on to really understand the mechanisms of this. So, and I'm a basic scientist, not a clinician, not an MD. So this is where my contributions are really a common thing. And the first thing I want to tell you is that these are very different things. They're not just two checkpoints and you can use them as immunotherapy, they're interchangeable. So I told you already, CTLA-4 is hardwired. Every T cell response is limited by that molecule, regardless of what it's against. PD-1 is an induced resistance that's caused by cells responding to gamma interferon produced in T cells. CTLA-4 targets the CD28 pathway, which means it works during priming. The very first time a T cell encounters an antigen, when it's being primed, as I showed you in the cartoon, that's when CTLA-4 works. We've done a lot of work in many systems now and shown that it also lowers the threshold of T cell receptor strength that you need to get activated, and therefore it recruits a lot of new clones into the response that weren't there earlier, that wouldn't have been primed if CTLA-4 had been around. It lowers that threshold, brings new clones in, and expands clonal diversity. PD-1 is an induced resistance, but it also targets the TCR pathway, and it works on fully differentiated cells. Again, they're trying to kill the tumor cell responds, and it uses this PD-1 molecule to try to protect them. So it does not expand clonal diversity. It does not bring new T cells into the response. Only CTLA-4 does that. So when you think about an immunotherapy and adding this, for example, to a vaccine, you're probably better off in adding CTLA-4 if you want to add a checkpoint than PD-1, because PD-1 will expand the cells that were already there, or those that you've primed that get to a certain point, but it won't bring any in from the start, whereas CD28 will. Dr. Sharma has shown in prostate and human and renal cancer at least, anti-CTLA-4 can drive T cells into an otherwise immunologically cold tumor, whereas PD-1 does not do that. If the tumor is cold, PD-1 blockade is not going to change it by itself into a hot tumor. Perhaps because the mechanism and the adverse events associated with CTLA-4 blockade are relatively frequent, much less so with PD-1, but also the disease recurrence after a response to CTLA-4 is quite rare, and I'll show you towards the end of this why we think that is, whereas they're less frequent overall in patients that are treated with PD-1, but disease recurrence after initial response are quite high as 25 to 30% in lung cancer and melanoma. And again, there are a lot of reasons for that, one of which I'll show you again. Anyway, these are given quite differently. Anyway, we wanted to look at the mechanisms involved. So we did CyTOF, a high-dimensional thing. I'm not going to go into the data, but basically we looked at 43 parameters at a time. It's cells that are being analyzed, and we included not only antibodies to differentiation markers on T cells, but also markers for proliferation, cell death, and other things that would let us know what's going on. And we kept track of tumor size in individual mice and treated just at the point, or took the samples just at the point when the tumors were beginning to shrink to analyze what's going on. And so these are the data. You can ignore this. This is a Treg data is an artifact of the antibody used in the mouse. This does not happen in humans. But in both humans and mice, what you see is that CTLA-4 expands upon, you see a big increase in the number of cells that are Th1-like cells. They have T-bet, they make gamma interferon TNF-alpha, so they're called Th1 cells. But they're a little bit different because they express a molecule called ICOS. Now, I'm talking about these now, and these came out of work that Dr. Sharma had done in a neoadjuvant trial much before we started this work. but she found these in humans, and you can see them here in mice. PD-1 does nothing to those cells, and these are naive CD4 cells. It doesn't do anything to those. But the number of these cells is associated with smaller tumor size. And CD8s, if you look at them, CTLA-4 does nothing, blockade does nothing to the so-called exhausted cells that you hear about that have PD-1, LAG3, and TIM3, 3 different inhibitory checkpoints on their surface. CTLA-4 does nothing to them, but PD-1 allows them, treatment allows them to proliferate. It doesn't change them though. They still have PD-1 and LAG3 and TIM3 on their surface. So when the antibody goes away, they're going to stop dividing. And so that's why CTLA-4 most of the time is given in a very small number of doses, whereas PD-1 can be given for up to 2 years until a patient progresses, because you have to keep giving it to alpha T cells to keep proliferating. So CTLA-4 does not touch those, PD-1 does. And those exhausted cells and untreated are treated with PD-1 correlate with smaller tumor size. There's another population of CD8s that's involved that are just standard CD8 killer cells. They have a little bit of PD-1 on the surface, but they are activated, fully differentiated, capable of killing cells and making cytokines. Both CTLA-4 and PD-1 expand those. The rest of the cells, immune cells, are irrelevant. Other types of CD8s than NKT cells don't really play any role in the response. So this is what it comes down to. With CTLA-4 monotherapy, you expand these CD4 ICOS positive cells and CD8 effectors. With PD-1, you expand the exhausted cells and the CD8 effectors. So the question came up, what happens if you put them together? That happened very quickly. Well, we put them together in mice, and they were additive. I showed you what happens in humans. There you get this big bump of durable responses, over 50% of melanoma patients. But what's the mechanism of that? So we also took a look at that by adding a 4th group in these, and that's CTLA-4 shown in purple. So again, if you look at those exhausted cells, which are the primary targets of PD-1, they're expanded not by CTLA-4, but by PD-1. But when you add CTLA-4, you don't expand them anymore. In fact, they go away. The numbers of them decrease. And in fact, they become irrelevant to the immune response. This is the increasing coefficient with cell number and tumor size showing untreated or PD-1, the number of those exhausted cells correlates negatively with tumor size, whereas if you add CTLA-4, they become irrelevant. The numbers go down and they become irrelevant. But what you can see here is if you look at those terminally differentiated effector cells, they go up even more. So you expand the soldiers that are really doing the killing there. And then if you look at CD4s, they also are increased more when you give CTLA-4 and PD-1, because they have PD-1 on their surface as well. So what happened with these exhausted cells? Well, it's been shown by John Wary and a number of other groups that there are epigenetic changes associated in the T cells the CD8 T cells as they develop, and they get locked in, supposedly to that triple positive that was an exhausted state, and can work only when the antibody's around, but they can't divide. So are we reversing those and turning them back into effector cells? We don't really know. We're looking for that. But I think the other more likely possibility is that they don't become exhausted if they have constant access to CD28 co-stimulation in the absence of CTLA-4. They just never become exhausted. So this has implications for how you treat. Right now, patients get CTLA-4 and PD-1 initially, and then they get maintenance with PD-1 for up to 2 years. I'm not suggesting that may change, but I am suggesting that be looked at a little bit more closely, because it could be right. This shows the expansion of the CD4s, or in that order, when you give the combination. So those CD4s are really important. But when you give the combination doses, you reduce it just to these 2 agents, the CD4 effectors with ICOS and these CD8 effectors. And what is ICOS? And here I'm going to, Dr. Shor's going to repeat some of these data, but I have to show it here. ICOS is a member of the same family of CTLA-4, CD28, PD-1, and even the B7 ligands that interact with CD28. It has its own ligand. But it's usually, if you tell an immunologist about a cell that has ICOS, they'll say, well, that's a follicular T helper cell, making IL-4 and other things, helping B cells class switch immunoglobulins. That's worthless in a perhaps worthless in the tumor. It's certainly not going to directly kill tumor cells. Or regulatory T cells, which turn off T cells, also have high levels of ICOS. That's not going to do anything. But Dr. Sharma, in her very first neoadjuvant trial of bladder cancer in 2006, identified these as one of the cells that comes up after treatment. I point out that that neoadjuvant trial was done 5 years before CTLA-4 was approved by the FDA. So you can do science and clinical things, but unapproved drugs and other things. So I'm not going to go through this in any detail. She will talk about this more. But it went to the mice and actually showed that CTLA-4 does not work very well in mice. And so anyway, we wondered where those T cells came from. And so again, back to basic biology, and wondered if perhaps there was something that had gone wrong or something different about the differentiation of cells. So we went back and looked for those cells in cells which genetically did not have CTLA-4. So we see these cells when we block CTLA-4 with an antibody, but the question is, if you genetically delete it, what happens? And so this is just the whole T cell population from wild-type mice in green, heterozygotes in blue, or those that lack CTLA-4 in red. And you can see those red patches are the new cells that are not found anywhere, from what we can tell, in mice or in humans, except when you treat with anti-CTLA-4 or when CTLA-4 is knocked out genetically. So there are these new phenotypes. And what are they? Well, just to show you, annotate them here. There are these 4 different types. This guy, you can see, is really messed up. To an immunologist, you can see that has 4 supposed lineage, dominating transcription factors. These cells express 4 of them. So these are probably intermediates on the way to fully differentiation of that stuff. But this population here is exactly the cells that Dr. Sharma showed were present in patients that are being treated. And incidentally, that was shown to be, if you didn't get these cells increased in patients, they did not respond to CTLA-4 therapy. You'll hear more about that from her. So these cells are created by the CTLA-4 treatment. They're not expanded from small cells there. This is something that when we start probing, as a scientist, I'm fascinated with this, because we start probing and giving a drug, we're not just expanding different cell populations. In this case, we're actually creating a new one that does not exist otherwise. These are the classical differentiation pathways, as you would see in most textbooks. The Th1s here, flicker T helper cells here. These Th1s here with T-bit, I want to make gamma interferon. I can tell you that these have ICOS, these have ICOS, these don't have ICOS unless you give CTLA-4. And these have it. And we think what's going on here is that when you take CTLA-4 away, the CD28 can more completely provide that co-stimulatory signal and give you a stronger response that's perceived downstream in the T cell, and you read out that it's got a stronger signal, and it takes away the limitations on the normal differentiation possibility. You get new possibilities that T cells can differentiate into when you take it away, one of which is that cell. And so we think that we need to rethink 2 things. One, we need to rethink T cell development. It's more variegated, it's more multi-step and not as dominant as we think, and perhaps a little bit more plastic. But also we need to consider the possibility that we've shown here that we're making the drug, the prodrug, the antibody is making a new cell, a drug, which in this case is an ICOS positive CD4. So this is an experiment that we did together with Dr. Sharma, showing that, well, this is an experiment showing that CTLA-4 is necessary. This is CTLA-4 therapy. ICOS is necessary for better, optimum CTLA-4 therapy. If you do the experiments in ICOS or ICOS ligand deficient mice, you lose a lot of the therapeutic effect of CTLA-4. And if you give an ICOS signal, this is a system in which the tumor does not respond very well to anti-CTLA-4 plus a irradiated melanoma cell vaccine. But if you put ICOS ligand into that vaccine, you increase the response rate from 20% or so up to 90%, just by giving the ICOS signal. And it's due to the signal, because if you do this in ICOS knockout mice, you don't get that implication of the signal. So this ICOS pathway is very essential to getting optimal therapeutic effect. And so we are now working on trying to find ways of making antibodies diagnosable and more effective. And so we then took a look at what's going on mechanistically by single-cell RNA data. I'm not going to show you the T-cell data because it basically confirms what I've already told you. We expanded the CD8 effector cells and those CD4s. But if you look at the myeloid cells here, again, these are just grouped according to similarities in gene expression. This area here are what are called M2-like or suppressive myeloid cells. They have arginase and other things. They suppress T-cell responses. These down here have class 2 on them and have thought of it as being more like M1 or cells that can help a T-cell response. And it's a busy slide, but what I want you to do is look just down here when you give anti-CTLA-4 plus the IFACs. Almost all of those inhibitory cells are gone. The only cells left are the myeloid cells that could actually help T-cell responses. So we just caused this global reformation of the myeloid cell population as a result of this therapy. So there's something going on by cytokine protection, other things causing epigenetic changes, we think, in the myeloid cells and bringing them around to where they can help T cells. And this is something that we need to explore more because I think we can get more power out of immunotherapy if we start paying attention to myeloid cells and find ways to regulate them epigenetically, to move them from the inhibitory kind into those that will support immunotherapy. And I think that's the next area of work that we're going to hear some more of that from later talkers, speakers. So the question of memory is very important. So it's become clear over the last decade that this has been in the clinic that the responses with CTLA-4 tend to be more durable. So we reduced this to an immunological paradigm. This just shows here we're using a tumor vaccine. If you immunize with this vaccine and then give either anti-PD-1 or anti-CTLA-4, you get these responses. And you come back and challenge the mice that have been cured later on. You can see here that this is if you gave a control antibody. These are naive mice. They grow here. The mice that were cured with PD-1 don't have much memory. Those that were treated with CTLA-4 now have full memory. They can completely reject that tumor. The ones with PD-1, not all mice do it. So to get the tumor out of the thing and do an experiment where you can rule out the possibility that they're just residual tumor cells providing constant challenge, we did the experiment with peptide. This also allowed us to get cells out, the exact cells that have been responding, and do analysis of them. But this shows the same thing. This is just immunizing with the peptide to ovalbumin with either CTLA-4 or PD-1, waiting 3 months and more, and then re-challenging with the peptide. And you can see the memory response here with CTLA-4 is considerably higher than that of PD-1, which is not much better than nothing at all. So again, the memory response is considerably higher. And we take, if we purify the cells from those mice, put them in, adoptively transfer them, and then treat other mice, we can see that we get a better response. Those cells are a lot more effective in eliminating tumor than those that were generated by PD-1 on a per cell basis. Those got identical numbers of cells. And then we go back and look at these 2 transcription factors that distinguish cells. TOX1, which tend to be in effector cells. TCF1 is in stem-like cells that tend to regenerate. We see that if you treat it with anti-CTLA-4, if you treat it with anti-PD-1, you get a lot more tox, which makes the cells perhaps more effective than you do with CTLA-4. But if you do CTLA-4, you get a lot more that have TCF1 here than with PD-1. And then so we got from John Waring mice in which these 2 transcription factors have been knocked out. If we knocked out the tox molecule, it really didn't make any difference on the immune response. PD-1 didn't have an immune response anyway, but you lose the you don't affect the immune response with CTLA-4. But when you knock out TCF1, you lose that increased memory then. The CTLA-4 is no better than PD-1 if you don't have that stem cell-like factor in the T cells. So CTLA-4 works, we think, by just taking those early cells that have a lot of TCF, expanding them, so that when you come back, they've got more of that. They're more capable of giving you a fresh immune response, a memory response, but cells that are more capable of generating the whole gamut of cells that you need to have function. And so that's one of the other differences, that CTLA-4 gives better memory. It's hardwired into that immune response, an inherent property of CTLA-4, which we can see played out with the durability of the clinical responses in cancer. And so this just summarizes what I've said there. Anyway, so just to end, we've got all these, since, again, CTLA-4 was the first inhibitory molecule, PD-1, the second one. We've got some positive checkpoints, ICOS being one, as I told you, 4BB, people have probably heard about. Additional negative ones shown here, LILRB4, LAG3, TIM3. The only one of these that's in the clinic is this LAG3, which is now approved and has been used in combination with PD-1. There are none of these other molecules that have been approved. And we think there are a lot of reasons for that. One of them is that in the pharma industry, you tend to not do combinations with something unless you've shown that it has efficacy as a single agent. But we know these things are not all there. And when you give one inhibitory molecule, like Sharma talked about this, others pop up. And so that this temporal thing, and so if you go look at baseline, try to make any decisions, you're not going to know what to treat with because these other ones come up. So we need to study the patients better. And also this family of molecules here are not expressed on tumor cells, but expressed on myeloid cells. And we found that these are also good targets for inhibitory molecule, blocking antibodies that remove the myeloid cells from being able, and also have some effects on differentiation of myeloid cells. So a lot of new work to be done, a lot more investigations needed. But what I just want to end with is that this is the way that oncology has proceeded in the past. You treat a large number of patients with a drug, and you see if you've moved the median survival over. The quality of care, that's fine. That's definitely, there's nothing wrong with that at all. Immunotherapy with CTLA-4, we showed you can do that and also get durable responses. This is sort of mimicking the melanoma story with ipilimumab. About 20% of the patients have durable responses. That goes up to 55, 52%, as I showed you with the combination of melanoma. If we added another one, maybe we could get that higher. But the goal now is not this anymore. It's this. It's to get that tail on the survival curve as high as we can get it, and as many different kinds of cancer we can do it by combining these therapies, as I said, with chemotherapy, with radiation, with surgery. There are neoadjuvant trials now that show that if it's added to surgery, you can get response rates of melanoma upwards of 70% in small trials that are still playing out. So the optimism that I have is that we can cure cancer, some of it, in some patients. Our job is to cure more of it in more patients, but we know how to do it. We know the basics now. We've got an inroad. I'm not saying we know everything. I'm saying we don't know enough. And so we need to keep doing the sort of work, the clinical work, sports with the basic work on understanding how these pathways work. And I think that we will get there. And if we can form a union, as I said, our institute, the Allison Institute, was formed to unlock the full potential of immunotherapy, and we look forward to working with you here at this August institution to do what I said, cure more patients. So thank you for your patience.
Well, thank you so much, sir. Words really fall short to express our gratitude towards you for delivering this very informative keynote lecture. As a mark of our gratitude towards the Honorable Chief Guest, Professor Dr. James Ellison, it is time for his felicitations. I would request our Chief Guest to please come into the center of stage along with the dignitaries. So your presence at our institution means a lot to the entire Mahatma Gandhi University family, and hence the felicitation has to be very special and full of warmth of Indian tradition. I request Professor Suri and also Dr. Nirmala Jagdish to join the dignitaries for the felicitations. First and foremost, may I request Professor Nirmala. For this felicitation, first and foremost, I would request Dr. Emil Swankar to present Dr. James with a floral bouquet. I request Dr. Vikas Chandra Swankar to felicitate Dr. Ellison with a shawl. And may I once again request Dr. Emil Swankar and Dr. Vikas Swankar both to honor him with a Rajasthani safa. And this safa signifies respect and recognition for any eminent person at the gathering. So sir, we are deeply, deeply honored to have you at this campus amongst us. Please give a huge round of applause. Next, I will request Professor Anil Suri to present this very beautiful memento. It is a kalash. As per the Hindu, from Kumbh, as per the Hindu rituals, it has profound significance and embodies the concept of abundance, prosperity and spiritual purity. It actually symbolizes divine energy and beliefs to sustain all life and nurture the essence of creation itself. So this very special memento and the conference plaque, I would request all the dignitaries on the stage to present the conference plaque to Professor